past would not likely have been detected had we not had the whole genome sequencing in this new technology. But what's really going to be the challenge for the future is it's only going to get better. And not that our food supply is less safe, it's just that we got better methods out there for picking these up. And so it's going to be I think, a real challenge for the industry to keep up with this. So unfortunately, our third speaker, Robin Anderson from USDA, couldn't be with us, but maybe you could share some insights, Peter and Noel. You know, what have we learned from the microbiome research to date that could help us do better in controls of harmful microbes that you can find in fresh foods, specifically, you know, produce and the meat supply and those types of foods. Any thoughts on that?
Well, it works. Okay. I'm aware that the metagenomics of food and the environmental is probably the area of that has moved forward more quickly than in the clinical area. So, but I don't know the details. I am a public health specialist. Do you know more, Noel?
I guess what I could say is that one thing that I think is sort of emerging from the literature and the research is that things that we're already doing to food are impacting the microbial communities and we don't know it. So I saw a research presentation a couple weeks ago where they talked about the microbiome of packaged meat, and the largest driver of microbiome differences was just the way it was packaged. So these are things that we may not think about as sort of manipulating microbial communities because that's just not the context that we package meat in, right? We're packaging meat for other reasons, but it is affecting the microbial community on that product. So I think one of the things we need to get a handle on is how do we define what we're already doing in terms of the microbiome, and how do we then say what we're doing is safe or not. So tying it to risk is, of course, the key. So
thank you for that.
Yep.
Another example that I've recently come across, a group from the UK as well as USDA ARS has been working on the microbiome of the chicken. And they've learned that when you feed soy-based products to the chickens, it increases the level and prevalence of Campylobacter, which is one of those foodborne pathogens often associated with poultry and one of those hard nuts to crack. So maybe we need to look at the diet a little closer than what we've realized in the past. Yes, sir.
First of all, thank you for some excellent talks. Really appreciate that.
Could you state your name?
Mark Freiling from General Mills. The question for the panel is, can you comment on our confidence around the uniqueness of a particular sequence to the environment where that sample was taken? So if we take a sample from a manufacturing plant or a feedlot and we sequence it, how confident are we that that sequence would never show up in another sampling environment?
Unless you know how it was introduced into your environment, you can't answer that question, can you?
So so maybe as a follow-up, are we aware of identifying identical sequences that have been taken from different sampling locations, from different, again, manufacturing plants, or from different, different locations where the samples were obtained?
I'd say yes, we definitely have that, because that is the reason why we still need epidemiology when we're doing outbreak investigations. Just because you find a match between a food or a food production isolate and a group of clinical isolates, that doesn't mean that that food is the source. It could be something else that has the transmission in common. The distribution chain for a microbe can be very complex. just as complex as the food and even worse, I guess.
Perfect. Thank you.
One thing I would just wanted to add to that too is that the uniqueness of sequences between microbes is not usually there. So many of the sequences that we get when we sequence these microbial communities are homologous between different species or strains or genera or even higher up in the taxonomy. So that's what makes it tricky to do this microbiome classification and to actually name specific organisms within the metagenome, because so much of their, and oftentimes so much of their genomes are homologous or similar or even identical. And when we're doing the metagenomics and we're fragmenting it up, oftentimes we get a piece of DNA that does match to multiple different organisms. Well, you can't classify that very well, right, with a lot of resolution. And so I can say that one study we did, for instance, we took feces and we looked at salmonella from culture PCR and shotgun metagenomics. And when we first did the shotgun metagenomic analysis, we found salmonella in everything, but the culture and PCR results said it was actually very, very rare. Salmonella was very rare in this sample set. And when we looked, what we were actually doing probably is, it gets a little bit in the weeds, but when you have a sample and you're taking all of the DNA and then you're trying to match it to a database, if you have organisms in your DNA that have never been characterized and are not in your database, but they happen to have pieces of genome that match to things that are in the database, our algorithms will say that those pieces of DNA came from the thing in the database because that's our only reference that we have, right? And so you have this problem where, like, we certainly have not sequenced and characterized and named everything that's out there. I mean, we've hardly scratched the surface. And so when you're using reference databases, you're biasing your results towards things that we know. And there's misclassification when that happens.
Could I follow up on the point that Peter made? As I understood, you believe that the epidemiology has to be linking to the whole genome sequence data in order to connect what comes out of a food plant or what comes out of a food to that's causing people to get sick. Do I have that correct? That you have to have the epidemiology to link the sick person to the food or the food processing plant through whole genome sequencing?
We are not going to take any action if we don't have the epidemiology.
If you don't have the epi. Okay. But let me share with you a recent example that's kind of this gray area regarding this. It's been in the news a few weeks ago where there was a recall and an outbreak associated with a unique strain of Salmonella agbeni. And it's a rare strain. I think there were like 6 cases of this particular type of Salmonella in the human database for 2018. There was an outbreak associated with this strain of Salmonellosis in 2017 associated with pet turtles. Mm-hmm. In any event, the scenario was one of the state health departments or agencies went out and tested at retail samples and found this particular strain of Salmonella in Duncan Hines cake mix. And this is public information. It was in the news. And so the whole genome sequence, as I understood it, linked the two. The Salmonella from the cake mix and from, I think there were 5 cases, human cases. So the whole genome sequence linked. But when you did the epidemiological follow-up, there was no match to Duncan Hines cake mix.
I'm not sure that is correct.
Well, I am. And then they found, there was another 6 isolates of Salmonella agbeni in the system later. And my understanding is they didn't match the Duncan Hines cake mix either. And the whole genome sequence was the same, but they did not match with the epidemiology of being exposed to Duncan Hines cake mix. And I know CDC and FDA were very careful in how they worded this situation in the data that was released to the public, but the press didn't do that. They said this outbreak is associated with Duncan Hines cake mix. So my point is, I think this is a learning experience where if we're going to try to get the industry to do more testing and in-depth whole genome sequencing, there has to be more clarity in how those results are released. I'm not finding fault. Don't get me wrong, Peter. I just think it's a good learning experience for everybody in terms of how whole genome sequencing can be used and is being used and how it's being perceived by the public and the press. Joe.
Thanks, Mike. Joe Sameka with Cargill. So I want to thank both speakers for really interesting and thought-provoking presentations. So I have kind of three comments that are somewhat interrelated, and maybe it's more directed towards Peter. But while I think it's good that we're measuring outbreaks and how better we're able to solve and attribute the cause of the outbreak, shouldn't we also be measuring public health impact? And the real measure shouldn't be public health impact. And I know it's hard to measure something that didn't happen, but still I think we can use past performance as a way to predict how improvements, prevented illnesses, and that we can determine to be a public health impact. So that's kind of one comment I have. Then secondly, as you pointed out, a lot of the time that it takes to really solving these outbreaks is collecting the data, identifying the outbreak, collecting the data, boots on the ground work. And largely that is done by the state departments of health. And they are chronically underfunded. And many of them have very poor effort that they put towards food public health, related to public health. They're worried about other issues like other public health impacts, and not enough epidemiologists worried about food and the food supply. And so what can we do about that? And then wouldn't it help to standardize the way in which we investigate those outbreaks, that they're done 50 different ways, different surveys that are used by the states. They each have their own ways in investigating these outbreaks. So shouldn't we also work hard to improve that, because a lot of the time that it takes is really upfront. And the last comment I would make is, if we're really going to improve the system, then when we do determine the attribution of an outbreak, shouldn't we really spend a little more time understanding the cause and effect of the outbreak? And that's really different. What really led to the contamination of the food? And then communicate that information more broadly throughout the system. We often solve the outbreak, in some cases punish the offender, and then move on. And we're not really benefiting from all that hard work and money and effort in getting the system to improve, because no one else is really learning from another's mistake.
Good questions. Peter?
Yeah. Well, the first question about the public health impact, we have actually been working on that. A couple of years ago, we published a paper on exactly that subject with the help of some researchers from Minnesota and an economist, and what we could document, and it's fancy calculations. I can't explain them in a simple way, and I'm not sure I quite understand myself everything. But in that study, we found that we had prevented about 270,000 foodborne infections of Salmonella, E. coli, or Listeria every year the past 10 years. And at the same time also saving the American society a cost of about $500 million, I guess, per year in medical savings and lost work. So that's definitely there. We expect that cost benefit is going to be even greater with whole genome sequencing because it's more precise. We don't know, but we plan also to repeat that investigation at some point. The other thing about how to make the outbreak investigations more efficient, I completely agree, and that we also need to standardize the epidemiological follow-up. And we have been working on that actually since about, I guess, 2010 or something like that. CDC has created, with the help of APHL and some federal funding, some sub-networks that includes the epidemiologists, the Outbreak Net Enhanced and the Food Core networks, where the purpose is for the states to combine the data from PulseNet with intensified epidemiological follow-up. And we know that if you do more efficient follow-up, you will detect more outbreaks and you're going to solve more outbreaks. Actually, one of the findings we had in the cost-benefit study we did with PulseNet, we did a correlation between the number of outbreaks that were detected and solved in the different states by how much they were using PulseNet. And the states that were using PulseNet were much more efficient in working with the outbreaks than the states that were not that active in PulseNet. So we are doing things, and we can definitely do things better, but it's also a resource issue. Then the question about the cause and effect of outbreaks. We are increasingly also doing more investigations where we are going back to the ultimate source and looking at how the ultimate source became contaminated and acting on that. Of course, CDC is not so much involved in this. This is more the food regulatory agencies, but I know that both USDA and FDA is putting a lot of focus on that at the moment.
Thank you. Noel. Got a follow-up question for you. What about the great studies you've been doing? Have you been able to identify any factors in your microbiome research that could lead to the reduction of the antimicrobial resistant bacteria?
Well, I think that things that we're already doing on the post-harvest side, some of those are reducing the bacterial populations and then also the resistance genes as we showed. But in terms of sort of on the live side, I think that there's a lot to be done to understand, a lot more to be done to understand how different things that we're feeding the animals or giving the animals is changing resistance, and that's a big I think emphasis that I'd like to convey is that when we talk about sort of alternatives to antibiotics, I think one of the things we should look at is how are those alternatives to antibiotics impacting the microbiome resistome, because they probably are in some way, but we need to look at it as an outcome. So,
Thank you.
Yeah.
Terrific points. Art?
Art Liang, CDC, and I'm at an age where I get to ask stupid questions, so and maybe the answer is, I need to go back to Biology 101. But to what extent, when we talk about resistance genes or the resistome, are we talking about correlation versus causation? And so that could be really a stupid question for most of the people in the microbiologists in the room. But so it comes down to, when we say this, genes code for proteins. So I guess it's the difference between saying we know the pathway from this gene producing these proteins and feeding into the mechanism of resistance that we know, versus we've identified a gene, and then when we test them, phenotypically test them against antibiotics, they correlate with this kind of resistance.
Yeah, that's a question that crosses from metagenomics also into whole genome sequencing, right? Does genotype match phenotype? And all I can say is in some cases it does, in some it doesn't. And the other thing I'll add in here, and I didn't talk about this technology, but we can also do metatranscriptomics. So we can look at the total population of transcripts in a microbial population. That's still not at the protein level, but we're one step closer. So we can add that as a layer to what we're doing. But so I work with a computer science physicist, and he always says to me, if people are stopping doing phenotypic work, like if that's sort of disappearing, he's like, that's really bad, because all of our big data algorithms and machine learning tools and all of these fancy words that we throw around, they all sort of, at some point, we need to validate them against, quote, he calls it the ground truth. I said, well, what's the ground truth for resistance? He said, well, it's an isolate growing on a plate that has antibiotics in it. And I said, well, is that really the ground truth? Is that isolate making somebody sick? Is it causing some, clinical or public health or a production outcome that we care about? And he's like, well, that's true. So, there's also the link, there's genotype to phenotype, but then there's also sort of in vitro phenotype to what's going on in the complex system, right, in the real world. And so I think that going through all of those links is really important at this stage in the game because it is a new technology. At some point, we might be able to just go from genotype to outcome, right, which would be great. And that's some of the things that you talked about as well. But at this point in the game, I think linking genotype to phenotype is something that we need to do to sort of historically contextualize some of these genome results that we're getting.
And I might add, it is biologically very difficult to correlate, if you're doing a metagenomic analysis, the resistance to the microorganism, because many resistance genes are on mobile genetic elements. And if you do a metagenomic analysis, you have to find a way to link that plasmid to that specific organism. You don't know if the plasmid you're looking at comes from a Salmonella or an E. coli or something third. And there are ways to do it, and we are looking into it, but it's difficult.
Yeah, we just did an analysis where we tried to bioinformatically link resistance genes to their surrounding context, whether that was a chromosome or a plasmid or mobile element. And you can do it, but I don't know if the results are really reliable. And so some of the long sequence technology might help in this respect, but it's just not high throughput enough yet for metagenomic samples. So that's something that maybe is on the horizon. Some technologies like ILSI, where you can do this genomic localization, might be promising. But right now, it's totally true. We cannot, with any confidence, say that this resistance gene is being carried by this bacteria within a metagenomic data set.
Okay, we're down to just a few minutes, so Richard, you have the last question.
Okay, well, first, thanks to Art for opening the floor for stupid questions, so I have one. And trying to follow what you are saying and what Mike mentioned earlier, we're getting an increase in recalls, basically, due to these new technologies, right? To what extent will even part of the recall be like a false positive for a specific product or a firm, or a false positive because we've misidentified something as a pathogen when in fact it is not?
If we are doing culture-based surveillance and use the epidemiology probably, then I don't think the number of false recalls is going to be very big. Of course, when we are assessing each outbreak, we also assess the organism and the impact that organism has. If it's a very dangerous organism, we need to act faster and on more loose evidence than if it's more trivial garden variety Salmonella, for example. But in general, we are pretty confident or I shouldn't say we, because we do not have any regulatory authority, but the food regulators, they would like to be on solid ground before they force any recalls.
All right. Well, please join me in thanking both Peter and Noel for just two excellent presentations. Thank you. Pierre?
Yeah, thank you very much. And it's just such a shame that we didn't have the